Good morning, everybody. It's really nice to see you all here. Thank you for joining us. I'm Nergis Mavlvala, and I'm the Dean of the School of Science here at MIT. I share President Kornblut's excitement and wonder at all that we have achieved in the life sciences at MIT over the past decades. MIT has been a place where fundamental knowledge and understanding of biology happens, where rapid conversion from ideas to discovery to translation happens, where computation and policy meet science and engineering comfortably, where innovation is a natural destination on the journey from discovery to societal impact. As well as we do all of this, as you heard from Sally, we know we can do better. And I couldn't be more energized to be here with you today to celebrate and dream of the future that the MIT HEALS Collaborative will help create. By catalyzing collaborations in fields like immunology, neuroscience, AI, sustainability, and healthcare, and strengthening MIT's ties with Boston's extraordinary hospitals and industry leaders in biotech and pharma, the MIT HEALS Collaborative aims to spur discoveries with broad and lasting real-world impact. Now, you couldn't come up with a better example that aligns with those ideals than our very own Dr. Phil Sharp. He has been at the vanguard not only of scientific discovery, but also MIT's collaborations with biotech, biopharma, the healthcare industry his entire career. Now, before I tell you a bit more about Phil, let me say a few words about how the MIT HEALS Collaborative came about. It does involve Phil, as many wonderful things that happen in the life sciences at MIT does. Little over 3 years ago, at the instigation of many wise colleagues and friends of MIT, and many of you are in the room here today, Dean Chandrakasan and I charged a faculty-led committee to study what we might do better to coordinate all the fabulous but disparate work in the life sciences being carried out at MIT. And also, how might we better nurture and curate collaborations with the life sciences research being carried out in the biotech and hospitals around us? That study, led by Professors Tyler Jacks and Christela Prather, culminated in the VITALS report. Now, VITALS stands for a vision to integrate, translate, and advance the life sciences. A key finding of that report was the hunger among MIT researchers for more opportunities to collaborate across disciplinary boundaries. The opportunities identified and the recommendations of the VITALS report were embraced by President Kornblut, and with the prodigious energy of our Chief Innovation and Strategy Officer, Dean Chandrakasan, here we are today, embarking on a truly vital endeavor. Now, upon hearing of the faculty's interest in collaborating with each other and with our award-winning hospital systems, the Kendall Square Biotech ecosystem, and beyond, Phil set to work helping us build those bridges. A pivotal moment for me was a gathering where a group of us were brainstorming about what this collaborative effort might look like and what big ideas would we lead with. Lots of ideas were being batted around, and at one point, Phil challenged the group. "What do we do best at MIT?" he asked. Everyone in the room must have had their favorite answer, but only Phil had an answer so succinct and fundamental and yet so universal for us here at MIT. "We measure," he declared. As a physicist, I can admit I have never directly peered into a microscope and seen a cell. But I do know measurement, and that resonated tremendously. Our obsession with inquiry, with observation, quantitative characterization, experimentation, discovery, and invention were all wrapped into those words. At MIT, we measure. It's part of MIT's secret sauce that Nancy Andrews alluded to in the video you saw at the beginning of our session. It's what makes me so optimistic about the unimagined discoveries waiting to be made when we bring seemingly disparate ideas and tools together through cross-disciplinary collaborations. This is the promise of the MIT HEALS Collaborative, and I'm very grateful to Phil for his clarity, wisdom, and encouragement as we embark on this endeavor. Now, for those of you who don't yet know Phil, let me introduce him to you. Dr. Phil Sharp is an institute professor at MIT in the Koch Institute for Integrative Cancer Research. He is also the chair of of the advisory board for the MIT Abdul Jamil Clinic for Machine Learning and Health, or J-Clinic. In 1993, he shared the Nobel Prize in Physiology or Medicine for the discovery of split genes. These are genes that the genes in DNA are not one contiguous segment of information as they are in bacteria. In fact, they're split. And that messenger RNA has to be processed by splicing before it can be translated into a protein. This fundamental discovery would alter the course of how we understood genetic disease and the development of potential treatments. In 2004, for this work, he was awarded the National Medal of Science. You'll learn more about that discovery from Phil shortly. At MIT, he joined the Center for Cancer Research in 1974, serving as the director from 1985 to 1991, before becoming the head of the Department of Biology for 8 years. He was also the founding director of the McGovern Institute for Brain Research from 2000 to 2004. Phil co-founded Biogen, serving on its board for nearly 30 years, as well as Alnylam Pharmaceuticals, a leader in RNA interference-based therapies, with Professors Bartel and Professor Emeritus Paul Schimmel and MIT postdocs. Phil is an elected member of the National Academies of Sciences, the National Academies of Medicine, the American Academies of Arts and Sciences, the American Philosophical Society, and the Royal Society of the UK. His accolades, his advisory board service, his awards, and most importantly, his 500-plus research papers are too numerous to recount in the time that we have. Even with our ample measurement skills at MIT, it would be truly hard to measure his impact on biology at MIT and beyond. Please join me in welcoming Professor Phil Sharp.
Wow, what a day. What a way to start a beautiful day. I couldn't be more excited. I couldn't be more enthusiastic about MIT, MIT Heals, and what it's going to mean for patients, people, the country, Massachusetts, and MIT. What to do in the next few moments is to give you a brief overview of a journey that I think tells you why this initiative is so important. I know I'm going to be looking backwards and setting the stage for what you're going to hear today, looking forwards. But I do think it illustrates why we're here and what we hope continues to come out of this great collaboration, of which I am sure it will. So I'm going to look back over 50 years. 50 years is when the Center for Cancer Research was established here at MIT, and I'm going to bring that journey to where we're at now, and I'm trying to emphasize what changes, what major initiatives had this impact. I have conflicts of interest. Alnylam is the only one that I will mention some relevant data to. Let's look back at the late 1950s. MIT's leadership looked at the Department of Biology and decided that in the future, biology at MIT will be gene-oriented, molecular biology, driven mechanistically, incorporating chemistry and physics into this marvelous discovery of the structure of DNA by Watson and Crick and Rosalind Franklin's data. And that led to the recruitment of Salvador Luria to MIT. Salva was a founder of molecular biology, the mentor of Jim Watson, and came to MIT in the late 1950s with other faculty and people at MIT to create what is the Department of Biology today. In early 1970s, Salvador Luria led a number of people at MIT to apply for a basic cancer research center here. This is after the National Cancer Institute Act in Congress, and leading that initiative was Luria and Dave Baltimore in molecular cell biology and virology, in Herman Eisen in immunology, and Phil Robbins in cell biology. Here in the early 70s, the pillars of much of what we're talking about in healthcare, molecular immunology, cell biology, was established here as a basic cancer center. And remarkably, the culture of the Cancer Center is a culture of MIT. It's a culture of collaborative interaction, of people sharing ideas, sharing initiatives. And what is shown here on the slide is the group of molecular cancer biologists in the Cancer Center, with Dave Baltimore peeking down at the left, Bob Weinberg over his shoulder, I'm up on the right side. But this was a collaborative group of sharing ideas and MIT mode of science. And it had an enormous effect. Dave Baltimore received a Nobel Prize in 1975, 1 year after the establishment of the Cancer Center. And then Susumu Tomogawa, who was recruited to the Cancer Center, shortly thereafter another Nobel Prize. I after that, and then Bob Horowitz at MIT, who was in the Department of Biology and a member of the Cancer Center, also received a Nobel Prize in the period of the Cancer Center in its site in the chocolate factory on Ames Street. I want to illustrate why this initiative of the Cancer Center was so timely. Here in the early 1970s, there was the beginning of the understanding that the pillars of biology, which was the central dogma illustrated here as DNA information flowing to RNA flowing to protein, had just been established by Jacob and Monod in 1961. And what you see here is a simple diagram that the RNA is being copied from the DNA in a collinear way, and proteins translated from it. And there is a sugar that is metabolized by these proteins, and that sugar regulates the expression of this gene. And this is how we understood molecular biology in the early 1970s. But we thought that the world was more complex than that when we started talking about multicellular organisms. Was the gene and how it was expressed the same in multicellular organisms? And there were hints, and I won't go through those, that there was something different. And that, for example, led to the understanding and discovery that genes in our systems have different structures. They are split, and they are expressed by an intermediate step of the splicing of the RNA, removing the dashed lines shown at the top of the slide and joining the black boxes. The black box is the sequences that code for the protein. So here, the complexity of the genome and complexity of how it's expressed and how it's regulated was much more complex than it was in bacteria, and it led us to understand that we were in a new territory of understanding disease processes. I also want to note the splicing pattern on those genes vary from cell to cell and condition to condition. And therefore, even though we have 23,000 genes, we have many more proteins. They vary between cells from those proteins by alternative splicing, and they carry out different functions. And we're just, in the last several years, able now to look at a single cell and say, this is exactly what the gene is expressed at in those cells. But also, in the same time period, in 1975, we had this convergence or confluence of remarkable discoveries that created synthetic biology, the ability to make organisms that had never existed before. And that was this recombinant DNA technology, DNA sequencing and chemical synthesis. So we could take DNA and we could make new genes and we can put genes in organisms. And this technology was recognized to be a transformational technology. And the first biotech community or company that was established in 1976, Genentech, was by Bob Swanson and Herb Boyer. Swanson, an MIT graduate, Boyer, as a scientist at San Francisco, led to this foundation of biotech and the whole biotech industry. And I had the pleasure of collaborating with Wally Gilbert at Harvard and 6 European scientists in establishing Biogen in 1978. The idea here is this technology had been developed in academic labs, and it wasn't going to benefit anyone until it was translated into the private sector, gain capital and investment, recruit people, and make a difference in people's life. Be innovative in this technology. And that led to Biogen being established in Geneva in 1978, when the situation became a little clearer in Cambridge as to what you could do with recombinant DNA. In the early 80s, this moved to Cambridge. And what I show you here is what Cambridge looked like in 1975. What you see to the right of that is Main Street and MIT across the street, but you see a large parking lot. A parking lot, for historical reasons, was open, and Biogen began in a little building on Binney Street on the left side of that slide. So here, Cambridge was the site in which this new technology was beginning to nucleate. Genzyme and other companies established in the same area shortly after at the same time. But then it grew and it consolidated and it collaborated, and that became the Kendall Square area in 2012, where that same space is now occupied by companies that are making marvelous advances in important ways. And as Sally said, it happened because of the great universities, the hospitals across the street with remarkable faculty and clinicians, and the New England finance and venture capital acceptance. This continued to grow. We're the most concentrated biotech community in the world. Life science, it's a remarkably dynamic, collaborative, networking community that is reflecting a very important part. And I forgot to mention, it wouldn't have happened if it wasn't from MIT engineers. MIT engineers allowed us to take these products and make new components of it. So all of this happened, and now we see what Cambridge is like. I should illustrate here in 2018 that large pharmaceutical companies, research stations, research institutes have moved into the community, and there's probably 50 or more small to medium-sized biotech companies. Biogen has grown, and Alnylam, which I'm going to talk about in a moment, and then Broad I'm going to talk about immediately, as additions to this community that empowered a lot of remarkable advances. So we moved to 2000, the turn of the century, and biotech took on another acceleration in the country because of the development of the Human Genome Initiative and the Human Genome Sequence. And it's interesting to think about the origins of that work. Whitehead was established in 1985. It was a new institute relationship to MIT. MIT was open to bringing new resources and new structures to being part of the community. At MIT, at the Whitehead Institute, Dave Baltimore recruited Eric Lander, who started the Genome Initiative. And that Genome Initiative then moved with additional resources to the Broad Institute, which is across the street from MIT, associated with MIT and Harvard, again, a collaboration. And that led in 2003 with an international initiative, highly influenced and led by the Broad Institute, to the whole human genome sequence. This was an accelerator because then we had in our hand the sequence of every gene, the sequence of the genome, and we could begin to integrate complex genetics and molecular tools into this identification of, or this technology, powerful technology of recombinant DNA. This allowed us to more rapidly translate our science and our technology into patients. And just to illustrate how that happened, or the consequence of that, I will speak about a new subject that appeared in 2000, and that is with the genome sequence in previous science, it was recognized that our cells encode another 500 or more genes that we didn't know about that encoded small RNAs. These small RNAs were called microRNAs. The initial discovery of the first was in 1993 out of Gary Ruvkun and Victor Ambros. Furthered that work. It was done initially with collaboration in Bob Horowitz's lab, and out of that came this discovery of microRNAs, and as you will note, that was recognized by a Nobel Prize in this year, 2024. This microRNA pathway is present in every cell. It was totally unrecognized until the 2000s, but David Bartel and others, and it has become a major regulatory, involved in cancer, involved in many processes. And then, at the same time, in 1998, there was a discovery by Farr and Mello of the fact that double-strand RNA, most RNA is single-strand in the cell, but you can see double-strand. And double-strand RNA from a gene will silence the expression of that gene in many organisms. Their work was done in the worm C. elegans, but it was soon recognized that that work interdigitated with the microRNA work, and this led us to an understanding that there was another biochemical system in the cell we didn't know. Andy Farr was a grad student here at MIT, but did the work after he left MIT. Using that work, and then with David Bartel and Phil Zamor and Tom Tushel, postdocs, and Paul Schimble's influence, it became recognized that this whole process of silencing a gene with RNA offered a new therapeutic modality. We could use the natural pathway in the cell, design a drug, siRNA, from the sequence, and then treat a patient if the modulation of that gene would have an impact. So that was just another way of possibly benefiting patients. And that led to then the establishment of Alnylam and the confrontation that the big problem here was delivery of the RNA. And we turned to engineers and other chemists to solve that problem. Delivery of RNA into cells is difficult because RNA is hydrophilic and your cell has a membrane that's hydrophobic. And therefore, you have to transport the RNA into the cell. But once it's in the cell, it'll enter the microRNA pathway and carry out, if it's complementary to the messenger RNA, destruction of the messenger RNA, and then be catalytic. So the big issue here was, could you introduce this RNA into cells? And that led to the development of lipid nanoparticles, which look like a virus. They have a cationic lipid of a certain type. Peter Cullis taught us how it's a Canadian scientist who made major contributions to this. Impacting the RNA into these nanoparticles would, with the right ionic pH and pK, give you then a change in conformation of those lipids as the particle enters the cell. In your blood system, you have a pH of 7. In the cell, it's nearly a pH of 5 in this endosome, and that creates this transition. That same technology, is similar technology, was used in the vaccination in COVID that rescued us from the pandemic. It has then been used to treat billions of patients. Here again, you see the collaborations between molecular discovery to innovation to the translation to benefit patients, and then other people using those insights to further solve a major societal problem in the pandemic. So that brings me to looking forward at what you're going to hear in the next several hours from my remarkable colleagues here at MIT. And that's what we've called convergence. We, with the discoveries of the structures of sequences of DNA in the genome, pathways in the cell, mechanisms by which the cell carries out communications between cells, we have the makings, not a complete understanding, but the makings of how the cell works and the components. And if we look back over time, that's a product of research from in the mid-50s, as recruitment of Luria, the establishment of the institutes, research advances, discoveries of multiple types, and then we are at a stage in which engineering can take those components and add to our tools and understanding these processes, adding AI and change how we can empower our scientists to benefit patients to solve problems. And that's what we've called convergence, and it's what MIT HEALS is a major component in making, and it is the future of life science, and it's the future of benefiting a large number of patients. The Koch Institute is one of the examples that have been established around it. You will hear more today about the Koch Institute and its technology, but its faculty are a combination on every floor of engineers, scientists, chemists, life scientists, chemists, and physicists. We are a very interdisciplinary, integrated institute. But if you look at the McGovern Institute in Brain Research, the Picard in Brain Research, the Reagan Institute, you see this integration of technology and molecular cell and cell biology and the whole premise of looking outward and impacting on patients. And it is a remarkable, and I should include the Reagan Institute, remarkable set of institutes associated with MIT. No other institute that I know of has been able to create this body of collaborative institutes in a community with such a powerful biotech community and hospitals that is looking forward, as our governor said, in making MIT, Massachusetts, keeping it an absolute leader in this space. So innovation continues. Here is just the number of companies over the last, from 2007 to now, that faculty in the Koch has continued to use technology to make discoveries in technology and translate to patients. And that is the continued engagement with society, changing the outcome of our patients in our country with this technology. So I am really grateful to have this chance to speak with you. Everything I've talked about is a we. Everything has been done by collaboration with many people, with faculty, with our students, our postdocs. I show you here reunions of my lab over the last 30 or so years. All of MIT and all of my colleagues have contributed to making this moment. And I am terribly excited about what MIT Heals means for MIT and for the country. Thank you.